Hi, my name is KRISTIN, and I'm a fourth-year PhD student at the University of Ottawa. And today I'll be sharing some of my PhD thesis research using geospatial methods to quantify co-dispersion of mercury sources and exposures in river otter. So I know wildlife and river otters isn't the typical subject matter of this conference, but I hope that some of the methods that I present here today you can find some utility in and have applicability to humans. So as we all know, mercury is a global pollutant of concern. So mercury is released in its inorganic form into the environment through both natural and anthropogenic sources. Once in the environment, mercury can be methylated to its organic form, which is its more toxic bioavailable form, and methylated mercury is a potent neurotoxin. Once in the environment, mercury can bioaccumulate in organisms and biomagnify up the food chain. This puts top predators, including humans, predatory birds, and fish, at greatest risk for mercury exposure. And the drivers of methylation of mercury are different in different areas. And so different factors that can influence the methylation of mercury to its more toxic form can include the concentration of organic matter or dissolved organic carbon. So higher organic matter usually indicates higher methylation rates. pH can also be a driver of mercury methylation. So lower pH promotes higher methylation rates, as well as temperature. Higher temperatures also promote higher methylation rates. So how do we monitor mercury in the environment? So typically we use Sentinel species. And so the Sentinel species that I'll be talking about today are river otters, particularly the fur of river otters. So river otters are really good sentinel species for a couple of reasons. First of all, they're piscivorous, meaning they eat a mostly fish diet, and they're top predators, so they're at the greatest risk for methylmercury exposure. They are sensitive to environmental changes, and most importantly, they have very small home ranges, they do not hibernate, and they do not migrate. That means the sources of mercury that they're exposed to are from their local environment. Lastly, they're widely distributed throughout North America, and so information that we learn about otters in one location can apply broadly across the continent. And so otters, or sentinel species, really serve as our canary in the coal mine. They give us early warnings as to what is the contaminant burden in the environment, and this is especially important for high fish consumers, such as First Nation populations, and so it can give us indication on when we should be further monitoring. So there have been a couple national mercury monitoring programs in Canada, and so this has included a couple studies by Depew et al. Looking at the common loon and yellow perch. And so here in these maps, we can see that in Western Canada, we have lower concentrations of mercury exposure, indicated by the yellow. And as we move east in Canada, Eastern Canada has higher concentrations of mercury exposure, indicated by the darker blue colors. More recently, there was also a study by Chatelet et al. Looking at little brown bats and big brown bats, particularly the fur of these bats. And we can see a similar trend if you look at the box plot here. In Nova Scotia and New Brunswick have higher concentrations or higher exposures compared to Western Canada. So from these studies, they have identified important drivers of mercury exposure. And so these have included land cover, including forest cover, barren land, and agricultural land, as well as the soil pH content and soil organic carbon content, as well as mercury deposition. Some other sources of mercury that have been identified but were not included in these studies include forest fires, hydroelectric dams, and industry. So there's a couple problems when we start to model spatial data. So the first being spatial non-stationarity. So this means that patterns and relationships can change unevenly over space, and this poses problems for our traditional ordinary least regression modeling methods. Particularly, it violates the assumption of residual homoscedasticity. And so here we can see in this linear regression plot, in an OLS regression, we'll try and fit one line through all the points, when really maybe our model would be better specified by fitting a couple different lines through different clusters. So this can pose some problems for modeling. The second problem is spatial autocorrelation. I won't go into detail about spatial autocorrelation today, just due to time, but it is a problem when we have self-similarity over space, which typically occurs when we have spatial data sets. A sample that you measure at point X is very similar to its neighbor. This also kind of wreaks havoc on our traditional modeling methods. So based on these, the research questions that I had was, what are the patterns of mercury exposure in Canadian river otters? And then secondly, what factors influence mercury exposure in these river otters, and how do these factors change across space? So our analytical methods, where we collected fur and whisker samples from the North American Fur Auction, as well as the Joint Oil Sands Monitoring Program. These samples were collected between 2013 and 2016. And total, we have just over 300 samples measured for mercury, and these represent 131 unique geolocations. So once we receive our fur and whisker samples in the lab, we use a mercury analyzer to quantify the total mercury concentration. Earlier I mentioned that methyl mercury is really the contaminant of concern. In fur, we actually find that almost 100% of the total mercury is methylated mercury. And so the geospatial methods that I used a Geddes and Ord's GI star to indicate clusters of hotspots and cold spots of mercury exposure. And then in the actual regression modeling, first you start with an aspatial regression. If you violate the assumption of homoscedasticity of the residuals, then you move to a geographically weighted regression. So I'll be presenting the GWR results today. And you can see the predictors and metrics that I used. So I used a variety of different predictors in my model. And so just to give you a little bit of a background on geographically weighted regressions. So geographically weighted regressions kind of decompose your study area into many local regressions. So you actually have one regression for every point in your data set. And so it uses a bandwidth or an area of inclusion, and for each sample point, you calculate a regression with its own beta coefficients and R-squared. So on to the results. So this is a map of fur mercury concentrations in Canada. So the larger the circle, the larger the concentration. So our concentrations range from about 1.3 ppm to around 50 ppm. So just to give you a little bit of context, we use 15 ppm as our fur mercury screening guideline. Above 15 ppm, we start seeing changes in brain neurochemistry of river otters, indicating that there might be some environmental problems. And so the patterns that we see, it's very similar to the other mercury monitoring programs in Canada. We have very low concentrations in Western Canada, particularly Alberta. And then as we move east, we see much larger concentrations, particularly in Northern Ontario and Nova Scotia and New Brunswick. So the hotspot analysis kind of confirms our visual assessment. We can see that in Alberta, we have a cold spot. So a cold spot just means that there's a cluster of low values that cluster together. And then conversely, in Ontario, Western Ontario, we do see a hotspot, so we have a cluster of high values. And then we do see a little bit of clustering over in Nova Scotia and New Brunswick. So I haven't presented all of the variables that I included in my model today for the sake of time, but I'm just highlighting a couple. So organic carbon content. So I'm just going to give you a little bit of context to explain these maps. So the yellow dots indicate not significant, and the darker the blue, the higher the significance level in a negative relationship. So taking you back to your statistics days, the map on the left-hand side indicates your t-values, which are directly related to your p-values. The darker the color, the more statistically significant. So we can see that soil organic carbon content is a pretty important predictor especially in Western Ontario and throughout moving east in Ontario, but not really a significant predictor elsewhere. This is also reflected in the map on the right-hand side, which is our beta values. So where it's most significant, we have the highest level of influence as indicated by higher beta values. Another important predictor is the proximity that these otters live to dams. So we can see that in Alberta and Western Ontario, this is a pretty important predictor. It's a statistically significant predictor, and then this is also reflected in the magnitude of the beta coefficient. We can see that it's most significant or most influential in Alberta and a little bit less influential in Eastern or Western Ontario, but still kind of important factors. And so I'm just going to map the local R-squared. So the local R-squared is comprised of all the predictors that go into the regression model. So other significant factors that were not shown were pH, the amount of wetlands around the river otter, and then number of forest fires. I'd also tried to include anthropogenic sources in the model, including MPRI, proximity to active mines, but these did not show up significant. So here our local R-squared ranges from about 5% to upwards of 50%. And so we can see that our model predicts relatively well in more Western Canada, but predicts pretty poorly in Eastern Canada. So just to wrap up, so in conclusion, drivers of mercury exposure in wildlife are not the same across Canada. This is especially important for very spatially heterogeneous landscapes, as Canada is. And these factors of mercury methylation are important regionally. And so this helps us identify localized patterns of mercury exposure, and this allows us to develop localized mitigation strategies. Applying one kind of blanket strategy across the country would not be applicable. And this study also kind of highlights how mercury is easy to sample, it's a minimally invasive matrix, and it can be good for a national mercury monitoring program. So our future work is to fill in some of the gaps for mercury data. As you saw, we're missing some provinces, so we're working to get data for those provinces and also to work to improve the model fit, especially in Eastern Canada. So one of the variables that I didn't include was soil mercury concentration. And we know that in Eastern Canada, particularly Nova Scotia, has really high naturally occurring mercury in the bedrock. So that's something to look to include to help specify the model a little bit better. So I just wanted to thank all my collaborators, especially the First Nation collaborators and all of my funding sources that helped to make this research possible. And with that, thank you.